Hi, everyone. Thank you so much for tuning in. We have three wonderful speakers lined up for today's topics on the neuroregulation of sickness behavior. And I want to introduce our first speaker, Jessica Osterhout. She is currently an assistant professor at University of Utah. And if you are interested in her work and her beautiful personality, which you are going to see very quickly, please contact her, don't hesitate. And we are going to have 25 minutes presentation followed by 5 minutes QA. We encourage you to raise your hand during the QA or type your question in the chat. And I want to, just because of the interest of time, I just want to mention one of the numerous awards that Jessica has received. She has received this Mel Selson Award in 2021, which is for the most beautiful experiment of the year. That pretty much sums up her work. Jessica, please take away and tell us your story. Thank you.
Thank you. And thank you so much for inviting me. I actually am so excited to present here and with the other speakers. I think it's gonna be really fun to hear all these different stories that sort of came out around the same time. So I'm really excited. The title of my talk is The Neural Basis of Sickness and Immune-Driven Behaviors. And when I say sickness, what I mean is this group of different symptoms that arise when we have some sort of flu or cold or some other kind of maybe chronic condition, and they include things like fatigue, loss of appetite, fever, even depression-like symptoms, changes in pain sensitivity, changes in social interactions. All of these things change at the same time, and the idea is that perhaps because they're sort of stereotyped and coordinated, that they serve some purpose for our ability to fight off infections. But what was interesting is when I started my postdoc, which was now quite a number of years ago, there was really no evidence as to how these different behaviors or symptoms arise. But what was clear from a neuroscience standpoint was that the brain was involved. So certainly, there's like this immune reaction that's happening in the periphery that, is important for, generating the immune response that actually fights the pathogens. But the symptoms themselves, other than things like, like your cough or your nasal symptoms, all the other symptoms are likely controlled by the brain, because the brain has these circuits for homeostatic set points for things like body temperature, appetite, things like that. So what areas of the brain are important for sensing the immune state, and which cells are important for generating these symptoms was a primary goal of my, well, I guess I should say it's a primary interest of mine then and now. So I've, as a postdoc, focused, just on a small piece of this pie. I focused primarily on the generation of fever. And the reason, actually, there's a lot of historical reasons that I could get into offline, but scientifically speaking, it's fascinating because it's heavily conserved by not just warm-blooded animals that have, brown adipose tissue that generates thermogenesis, but also by cold-blooded animals who use a behavioral mechanism. Like iguanas, for example, they will move to a warmer environment the same way they would do if they're trying to digest their meal. They would do that when they're infected with a certain pathogen in order to generate this higher body temperature. And now we know actually that this higher body temperature is really critical for activating certain kinds of immune cells. So now people have put immune cells into a dish and put them at warmer temperatures that are similar to a fever, and shown that those cells, some of them at least, become more active. They have a more active-like state. They're generating more molecules that are important for activating other cells. So fever seems to be really important for our immune response. And in addition, it's highly, highly conserved. So there's likely these hardwired mechanisms in the brain for generating fever. So that was the main question that I focused on in my postdoc, was how fever is generated and how it might be related to other sickness symptoms. So I just want to briefly tell you about the model that was in place at the time. And so when I had started, there was this paper that was like, about 10 years old, showing that this model where the preoptic area of the hypothalamus is pretty important. And we now know that actually this area of the hypothalamus, if you inject things like, different kinds of immune molecules, you can elicit a fever, but the neurons were really unknown. So the hypothesis or the model was that in the periphery, where pathogens are encountered, they activate immune cells, which generates all these cytokines. And cytokines are just, this large family of molecules, one of several that activate immune cells and other kinds of systems that are in place for fighting pathogens. And then the idea was that certain specialized epithelial cells in parts of the hypothalamus and other areas these are called circumventricular organs. And these are basically areas where the blood-brain barrier is sort of, people like to say it's leaky, so to speak. And what that just means is that the cells are specialized. So they have extra receptors, they have extra transporters, and they can move things from what's sort of like the peripheral circulation into the brain in sort of a regulated manner. And so these epithelial cells, and this includes endothelial cells that line the capillaries, as well as ependymal cells, which line the ventricles. These cells are specialized, and in this region of the hypothalamus, they have receptors for cytokines. And when they sense a cytokine, they generate something called prostaglandin E2. PGE2 was this molecule we know if we inject in this area, we can elicit fever, but it also generates a bunch of other sickness behaviors. And the idea was that PGE2 was then generated by the epithelial cells, propagated by microglia, and then sensed by local thermoregulatory neurons. And so the idea was that thermoregulatory neurons, although when active, they actually reduce body temperature. The idea is that they're repressed by this mechanism through one of the PGE2 receptors called EP3. And so then by repressing those, you get this change in set point that elicits a fever. So it seems like a fine model. And they do express EP3, it turns out. We found out a few years later when the thermoregulatory neurons were identified. But this model leaves out a couple of key things. One is the presence of cytokines in the brain. And so there was mixed literature at the time about what cytokines are doing in the brain. Are they even generated there? Where are the receptors at? And that's partially because our ability to sense or see them, with in situ at the time, we're not sensitive enough to even see their expression. So cytokines are expressed in the brain, and they actually have receptors on neurons. So where do those fit in this model? Are they even important? What about activated cell types? So we know that when we inject things into the mouse to give them this big inflammatory reaction, we get a lot of activated cells in the brain, and even close to the same region. So do those cells have a function? And then what's the relationship to other sickness behaviors? It's actually really not easy to separate changes in body temperature and other sickness behaviors. You have to use really specific manipulations to do that. So is there some sort of combined or some sort of mechanism where fever is sort of in the same circuit with other cells that generate other sickness behavior? So these are sort of the questions I had while reading this literature. And so my goal was to identify the cells and the circuits and the molecules that are required for generating fever and possibly other sickness symptoms. And so the model that I use, which you may hear about at least one more time today, is this model where we can inject something called lipopolysaccharides. It's an IP injection into the mouse. Lipopolysaccharides are just ground up bacterial membranes, and it's actually the bacterial membranes that have all these different proteins that your immune system senses. So we're giving them an immune response without actually giving them a pathogen, which is what iCook really loves. So we're going to give them LPS, and about 2 hours later, we see this influx or modulation of different symptoms, including body temperature. And so this is actually kind of challenging to do. And because you're going to hear about body temperature again, I want to just tell you a little bit about how we got an increase in body temperature, which actually is hard to do for mice. They're really small. So it's easy for them to, it's harder for them to increase their own body temperature, especially without moving, because, the sick mice don't move so much. So the way that we did this was first by doing a series of different doses of LPS, but then also paying attention to some of this literature saying that if you put mice at a slightly warmer temperature, they call it thermoneutral temperature, which is like, supposed to be where the temperature outside of the body is similar enough to the inside to where there's not a lot of heat exchange. If you put them at thermoneutral, it's much easier to get a fever in mice. So I did this, but the problem is that you also activate thermoregulatory neurons when you do this because they are warm. They don't actually want to be at that high of a temperature. So the trick then was to think about, how do mice generate a fever in the wild? Actually, like the fever is a real thing, so they must be able to do this in the wild. How do they do that? And so I finally was, thinking about this for probably too many months and realized that in the wild, we actually had some people across the hall that work with wild mice. And so we heard a lot about like the conditions that they use. So wild mice, they don't sit in a cage with a bed, they don't have this like white bed that they sleep on. They sleep in a nest. They make these, elaborate nests inside holes or wherever, right, that cover their entire bodies. And so I took advantage of the nesting material we had in our facility, which is not just the white nestlets you might be familiar with, but also this like brown cardboardy paper-like material. It's like shredded up. If you don't have that, it's mice love it. But if you give them both and a lot of it, what ends up happening after a few days is they make basically this nest in their cage. It takes up about half the cage, but it covers their entire bodies. And so when you give them this and then give them a sort of a lower dose of LPS on the wide spectrum of LPS you could use, they will hide in that nest and over the course of 2 hours have an increase in body temperature or a fever. So sorry for the long explanation, but I thought it would be important. So we can generate fever, right? So it took way too many months to figure this out. We can now generate fever, but we also look for other kinds of sickness symptoms. And we measure things like temperature preference. So just like you and I, mice tend to feel cold when they're sick, and they'll move to a warmer spot if you give them a long gradient of temperatures. This is not a huge difference, but it's very consistent. So they'll move from about 31 degrees Celsius to about 33 degrees Celsius. Imagine it's enough degrees to be like a warm blanket. And they'll have a change in appetite. They have a decrease in appetite. And then finally, I tested just general locomotion. So putting them in a novel cage, typically mice like to move around a lot in a novel cage environment to explore, and they moved around quite a bit less. And this is just a few of the different things we tested. I also tested changes in social interaction. I came from Catherine Dulaac's lab, who was very interested in social behavior. And if you've ever done social behavior, you know that there's a lot of combinations we can do, right? So different sexes, different ages, different environments. I like to use a social assay where there's no cups involved. They just are free to interact in the cage, and then quantification becomes more complicated. But basically, I found what most people have found, which is that if you have a mouse who's injected with LPS and a healthy mouse in the cage, there's an avoidance. And that's actually coming from both sides. This quantification is time spent interacting by the sick mouse. So the sick mouse is actually avoiding the healthy mouse, as well as the opposite. Usually healthy mice avoid sick mice. And there's other papers on that. But something that's interesting, and I don't even know why I tried this now, it's been a number of years, but I also tried other combinations. So these are male mice, by the way. I also put 2 mice in the cage that were the same condition. So either injected with both saline or both LPS, and found something really weird. And this is something that I didn't expect. And I thought I did it a couple of times because I thought I'd done something wrong with the LPS dosage or something. But what I found was that they actually had an increase in the time they were spending in close proximity. And so this is just, time spent in close proximity is measured with like a MATLAB script, like how often are they like next to one another. And there's a dramatic increase. And I can tell you, I don't really have time to talk about this story today, but I can tell you what's happening is that these 2 mice are doing this huddling behavior. And we know now that this is not related to warmth seeking. We can separate warmth seeking behavior from the social interaction. And again, I wish I had time to talk about this today, but anyway, so this is a story we're sort of wrapping up pretty soon. But to get back to fever and fever generation, I wanted to find the neurons involved in generating fever. And so I did what a lot of people in the lab did at the time, which was a CFOS screen. So looking for the protein expression of a gene called CFOS allows us to look at cells that are recently activated by, whatever stimulus. And this is about they have like the antibody is around for about 2 hours. So I look, at the 2-hour time for LPS, I look for any cells that are activated or expressing CFOS. And unsurprisingly, many areas are activated in response. This is in comparison to saline. So saline injection as a control because it is quite a stressful, thing for them. And all these different areas were significantly activated. But one of them in particular sort of stuck out, partially because of the location. So the ventral medial preoptic area is what it's called. It's in that same preoptic region as the thermoregulatory cells, and the same preoptic region as that circumventricular organ. And actually, I don't know if you can, I hope you can see my cursor here, but actually in the ventricle here where the ependymal cells live, you can see CFOS expression in the ependymal cells. And this is something that only really happens near a circumventricular organ. And the cells themselves here are on the bottom. There's about 1,000 of them, and they're very close to this midline, and then the base of the brain, which also has a bunch of different, or I should say, important cells. Anyway, so these cells seem very interesting. They're all inhibitory. So I want to know, what is the function of these LPS-sensitive VMPO neurons, and do they have any connection to fever generation? So to test that, to functionally manipulate these cells and test that hypothesis, I used a genetic tool called the TRAP2 mouse line. This is just a way to use Cre recombinase to target behaviorally relevant cells. So basically this inducible Cre is under the control of the CFOS gene. So anytime a cell is recently activated, this Cre is produced. And then if we add tamoxifen, the Cre is then translocated into the nucleus, where it can act on TD2 or any other actuator in the cell. So this is a super useful tool for anyone who's doing a long-acting stimulus. It's not, it's a little bit more backgroundy for things that are short, so like a short behavioral stimuli or something like that. But for long-acting stimuli, it's extremely useful. And what I found was I could, basically see that the LPS sensitive cells in the VMPO, if I use this tool, as opposed to what we call a saline trap, where saline is the stimulus. And so using this, I could target those cells specifically. So I had no need for like a genetic Cre, and actually we do have genetic Cres now, but even those are not as specific as this TRAP2 mouse line. And so I chose to use, in this case, a chemogenetic method. And the reason is because chemogenetics also act on sort of an hours type scale versus optogenetics, which is quite quick. And so I used this activation DREADS receptor and expressed it specifically in those VMPO cells and injected CNO. And so these controls are all injected with CNO here. But you can see when I use that chemogenetic receptor, we get an increase in body temperature, very similar to the febrile temperatures we saw with just LPS alone. So it seems that these cells are important for the generation of fever, but are they actually required for the generation of fever? That was the next question. And so for this, I used actually sort of a sledgehammer approach, where I actually just ablated those neurons entirely using a virus that expresses diphtheria toxin, and I can again express that specifically in those neurons, ablate them after 4 weeks, and then inject LPS again. So these are all injected with LPS. You can see a nice high body temperature in the controls. But in the experimental mice, where I confirmed the cells were deleted, we no longer get this increase in body temperature. And I honestly repeated this so many times because like this is really true. I have to be sure, and it certainly is. So it seems that these VMPO neurons are actually critical for the generation of fever. But what about other sickness behaviors? I told you I've now measured a number of these, and I went through all of those assays with the same experiments, and found that I indeed get an increase in the temperature preference of those mice. I got a decrease in the amount of chow consumed, but there was no change in the distance traveled, which is quite actually interesting that you can, kind of separate these different sickness symptoms in a way. But I also tried the deletion experiment and found that I lose the normal increase in preferred temperature, but I actually found no difference in the amount of chow consumed or distance traveled. And the amount of chow consumed was interesting, but I'm not actually surprised, which I can talk about towards the end. But it seems together that these VMP-LPS neurons are important for reward-seeking behavior, like the critical for reward-seeking behavior, and have a role in changing appetite during inflammation. So how do they do that? How do they modulate body temperature and feeding behavior? So with other controls, I found that these neurons themselves are not part of the thermoregulatory circuits. They're not activated during cold stimuli. They're not activated during warm stimuli. So they actually are not part of that normal homeostatic circuit. So how are they modulating body temperature? To get a clue as to how this might work, I did some tracing experiments where I injected the cell bodies with a tracer that goes along the entire axon, not a tracer, I should say, a virus expressing GFP that goes along the entire axon, and found that, again, like anywhere in the hypothalamus, they project to a ton of different areas. But two of these were really, really interesting. One is the median preoptic nucleus, and this area is important for things like osmoregulation, as well as other sort of homeostatic things, but importantly, it's important for thermoregulation. So this is the area where the thermoregulatory neurons were identified about a year into my postdoc. And so we knew what they expressed. We knew the genetic markers. And then the other area was the arcuate nucleus, which has long been studied for its controlled appetite as well as metabolism. And you can see projections to the more medial area of the arcuate nucleus. And so these are areas that were of high interest, as you might imagine. So the question was then, do the VMPO neurons modulate body temperature and appetite through these connections to those circuits? So to do this, I did a series of 2 experiments, basically. So I'll describe them. The first is a retrograde rabies tracing experiment. So rabies tracing is a way to look at the inputs of a neuron. The rabies virus is sort of truncated so that it can't jump to any synapse it wants, but if we give it the piece it needs, it'll jump just one synapse up. So what I did was I targeted those warm sensitive thermoregulatory neurons in the MNPO, gave it the viruses that they needed, and then looked for the inputs that were one synapse up. And we saw all the areas we were supposed to see, but that included the VMPO. And in magenta here is the LPS-induced CFOS expression. So you can see my cells here at the bottom, and then in green are all of the input cells. And you can see that those include some of those VMPO cells. So the VMPO is indeed forming inputs with thermoregulatory cells. But are they actually functional? So here I actually chose to use optogenetics. And the reason we did optogenetics here is because we can target not just the VMPO neurons specifically, but we can target just one set of their projections. So removing the activity of cells that project to other areas. We know that they have just a single axon that have not bifurcated. So I can activate on just the MNPO projecting cells. And then this time I used an optogenetics virus, not the normal sort of channel rhodopsin, but something called a step opsin, stabilized step opsin. And this means that one pulse of light will actually open the channel for about 30 minutes or maybe slightly shorter, but then the decay is about 30 minutes. So when I did this, and I chose to do this partially because that area has a lot of cells that are actually sense temperature, and by doing repeated optogenetic, pulses, we actually are increasing temperature of the area, which we didn't want to do. But it also is longer acting, which is again, I've tried to recapitulate sort of the normal physiology here. So when I did this, I used the same mice here, and we're comparing off trials and on trials of the same mice. You can see that with the single pulse of light, we're able to increase body temperature that slowly decays over 30 minutes. And so what this means, though, is that the VMPO is actually forming functional connections with those thermoregulatory cells in the MNPO, and the result is an increase in body temperature. And I did the same set of experiments with appetite controlling neurons in the arcuate nucleus, targeting those again and looking at the inputs and finding overlap with the VMPO cells and finding those are functionally significant. So activating just those terminals decreases appetite drastically. And I should say that I also used the same set of mice for the opposite behavior experiment and found that they don't have effect on one another. So activating the MNPO has no effect on appetite, and activating the arcuate projections has no effect on thermoregulation. And so it seems that the VMPO is then forming these functional connections with what we now know as the AGRP neurons in the arcuate nucleus, and that the overall effect is a decrease in appetite. And so, like I said before, there's a ton of different brain areas that were also activated by LPS. And I wanted to know if any of those might be in a downstream circuit to the VMPO. And this is a challenging question because the VMPO is inhibitory, which means any downstream target is actually inhibited, and we can't look at like CFOS overlapping with a marker to see if they're in the same circuit. But what we can do is look for any area that's disinhibited. So here's just sort of an example of what I mean. So for example, if the one area is inhibited, but they are normally inhibitory themselves, that means that their downstream targets are disinhibited. So when looked through all those different areas and found that a couple of them are in fact disinhibited in some way. One of them is called the DMH, and the DMH is involved in lots of things that have to do with homeostatic circuits, including thermoregulation. And in fact, we find overlap with 2 of the thermoregulatory cell markers there. And then the arcuate nucleus was also a hit, which was interesting because I just told you it was inhibited. But the arcuate nucleus also holds or contains neurons that have the opposite effect. The POMC neurons and CARP-PT neurons, their effect when they're active is to reduce appetite. And so those cells were also overlapping. So altogether at this point, what I found is a circuit mechanism whereby these VMPO neurons are able to coordinate the induction of fever and loss of appetite. But the question sort of remained, is this location important? How are these neurons activated? You'll hear today about other kinds of ways that neurons are activated during an inflammatory response. But here I was really, really interested in what's called the humoral mechanism of activation, where perhaps the cells are being activated through this CVO that's nearby, and through sort of a paracrine mechanism where there's sort of signaling happening directly to the neuron that has nothing to do with electrical activity. So to try to address this, I use something called spatial transcriptomics using a technique called MERFISH. And so MERFISH stands for multiplexed error-robust fluorescent in situ hybridization. It's a long name, but what it means is that we're basically doing an in situ hybridization where we're looking at mRNAs. We're looking at over 150 different mRNAs at one time. What that allows you to do is actually identify cell types based on the genes you choose. So we can identify cell types. We can also look at some of the things that they're expressing, and we can do this in a way that's sometimes species we normally would miss. So lowly expressed transcripts are easier to see. Anyway, so it's a really powerful tool, but not just this. So we're able to look at the gene expression, but in relationship to the spatial organization, which you can imagine is pretty important for this question if we're trying to look at the location of the neurons versus the CVO region. So here's just an example of one of those sections. In yellow is actually some of the warm sensitive neurons that are nearby. And you kind of like bleed into this area. And blue is the cluster of cells that was highly activated based on the MERFISH analysis. And in purple are those cells within that cluster that are CFOS positive. And you can see that those are seem to be towards the base of the brain. And we can, using this technique, quantify this and find that they are more likely to be CFOS positive towards the base of the brain or the midline. So something else that was interesting, sort of the first pass kind of experiments that we do, what we do in MERFISH or a spatial transcriptomics experiment is break down the cells that we see based on cell type, major cell types. So not just neurons, but also non-neuronal cell types. And so this analysis actually led to an interesting result, which is that the ependymal cells and the astrocytes also appeared to be highly activated, significantly activated. This is something we didn't see at all with other sorts of assays like aggression or mating assays, which we had also been doing in the lab at the same time. And so we looked at the organization of those and again found that those that are CFOS positive were closer to the base of the brain and closer to the midline. So this led to this hypothesis that perhaps VMPO neurons are activated by local immune signals that are coming from the ependymal cells or the astrocytes. So to get a better handle on that hypothesis, I did another kind of analysis called single cell sequencing, where I detected out just that region alone and then separated all the cells based on their transcriptomics. I could identify my cluster here, cluster 19 was the most CFOS positive out of those. But in addition, I could look really, really specifically at the expression patterns within the ependymal cells and within the astrocytes. And what I found was that those cell types, those ependymal cells and astrocytes, do in fact express receptors for immune molecules. They express the enzymes that generate immune molecules. And so these are just a few examples. So PTGS2 is an enzyme that generates PGE2, which I talked about at the beginning. And in green here, you can see it's expressed by the endothelial cells lining the vasculature in this region. And it's also expressed by some of these microglia as well. And then interleukin-1 beta is one of the most highly studied cytokines, and it's generated by these ependymal cells lining the ventricle, as well as these cells at the base of the brain, which there's several different cell types here, but including things like meninges and things like that, which are also immune activated. And then chemokine ligand 2 is another that was expressed by the ependymal cells, as well as I think astrocytes in here. So these molecules are not produced at time 0 or in a saline injected mouse. They're only produced after LPS at this 2-hour time point. And actually their expression patterns drop off soon after that. In addition, the receptors are expressed in that area, including, and some of these are a little more specific than others, but each of these are also expressed by the VMPO neurons. So I thought maybe this is a mechanism by which those neurons might be activated. And so to test that, I collaborated with a really, really fantastic electrophysiologist down the hall to look to see in a slice physiology experiment if those molecules had any effect on the activity of the neurons. And so he did a number of analyses, which I'll just kind of show you briefly. If you look at the firing frequency as with at different currents, you see that each of these molecules, as a function of currents, you'd see each of these molecules has an effect on the firing frequency. They're activated sooner and at higher frequency at the same current. In addition, we looked at peak firing rates, and although CCL2 didn't have an effect on peak firing rate, PG2 did. This was abolished by the addition of an antagonist for its receptor, one of its receptors. And then IL-1 beta had an even higher effect on peak firing rate, which is further increased by the addition of PG2. Excuse me. And then we also looked at the local synapses, so the synapses are forming connections with the VMPO neurons, and finding that, again, there was never analysis, and this is just sort of a summary. If you look at the excitation to inhibition ratio, so the ratio of vesicles coming from the excitatory inputs versus the inhibitory inputs, you see that there's a net excitatory effect by each of these molecules on VMPO neurons. So together, we found this mechanism where VMPO neurons are generating these sickness symptoms, and they're doing it through this paracrine mechanism where they're being activated directly by immune molecules through direct expression of their receptors on the neurons as well as on their local inputs. And the effect is that the VMPO neurons are able to sense sort of the global inflammatory state and induce multiple sickness symptoms at one time. So back to this sort of pie, we've now sort of figured out a small part of it, how fever is generated, but the question sort of remains, how are other sickness symptoms generated? And so I'm now I'm in this empty office in my own lab, and this is sort of the main question we're thinking about. How are other sickness symptoms generated? And so you're going to hear today about some evidence for things like appetite, and locomotion and thirst, but what about things like pain sensitivity or fatigue or depression-like symptoms? These are all things that are still sort of really poorly understood at a healthy mouse or individual, but how are they actually influenced during inflammation is a huge question that I'm very interested in. So I'm also interested not just in acute infection and for these different behaviors, but also in other kinds of immune activation states. So how is sort of this chronic fatigue elicited in a chronic inflammation model or something like long COVID, for example? Pain sensitivity is very interesting to me because it's not just affected by acute or chronic states, but also affected differently in males and females. So this is something that our lab is going to actively explore, are these different inflammatory states in the context of these different sickness symptoms. And so with that, I just want to acknowledge my current lab, which is small but growing. We have rotation students coming this year, but we're definitely looking for postdocs and technicians. I have a number of previous mentees that have worked heavily on different parts of this project, mostly on the social behavior project. Obviously, I collaborated with people in Catherine's lab, and I also want to acknowledge Catherine was a great mentor during this time, and then other collaborators on this project, and then funding sources, of course, that have been instrumental to my success. So thank you.
Thank you so much, Jessica. That was beautiful work. We have already one question in the chat, which I'm just going to describe it right now. The question is, do you look if there was any correlation with a preference to hot temperature environments and the survival? And the person who asked this question say that she's working with brain tumor bearing mice, and sometimes they see that those mice will get really cold before dying, and she assumed that those mice would also have a preference for hot environment. She's just curious about whether you see a preference with the hot warm-seeking behavior versus the survival rate.
Yeah, no, it's a great question. So what happens is that before death, there's actually a drop in the drop in body temperature is very normal for like a sepsis-like model. And certainly before animals die, they do get very cold. This is really normal. And I'm not surprised that you're saying this at all. Those mice, part of the reason they're cold is one, because their thermoregulatory system's shutting down. So they're not like, their brown adipose tissue is just not being activated. But I think those circuits that related to brown adipose tissue thermogenesis and warm-seeking behavior are linked. And so they're probably not seeking warmth either. I myself have not, so I focused on dosage of LPS where we get an increase in temperature because that was the goal of the project. So I never tested mice where I give them a higher dose and they got more of a sepsis-like effect with the lower temperatures. But I think also when they're close to death, their locomotion is even further decreased. They have even further decreased motivation to survive. They're not looking for food. They're not looking for warmth, probably. But yeah, I think temperature is a really good indicator actually of how a mouse is doing. When you see low temperatures, they're really not doing well.
Okay, can I just quickly follow up one question? I think one of the strengths of the paper is really you nicely line out this molecular mechanism with specific pro-inflammatory cytokines, receptors, and where they express, and all the way to the full circuit. And I was just curious about, is it local sort of propagation of the signals? And do you think that since those neurons also express the receptor for the cytokine, what is this sort of indirect, like, I don't know whether it's emphasized on the signaling at local in the brain. What do you think different cytokines, sorry, the question is getting a little bit, the different cytokines, are they involved in different behavior, or do you think it's the timing of those expression of cytokines relevant to the induction of different behavior?
Like why the combination versus,
right.
Yeah.
Yeah.
That's a good question. And so I'm learning a lot about immunology. But what I've learned is that during an inflammatory reaction in the periphery, immune cells will never be activated by a single molecule. It often requires two or more. And you can think about this as like a sort of a safety mechanism, right? Like you don't want to have a full-fledged immune response, because actually too much of immune response is bad, right? You don't want to have a full-fledged immune response unless you're sure that it's time. And so this mechanism of sort of multiple signals has developed in our immune system. And so I sort of, when I learned this, I sort of imagined this might be the same case for these neurons, that they have sort of a similar, I need two signals to be activated. But as you can see through electrophysiology, that one seems to be enough. Although these are, in slice culture, where like, it's not exactly like physiological levels. So it's really hard to say. But I don't think it's a coincidence that cells, the immune cells in the periphery also express multiple kinds of receptors and respond to require multiple signals. And we're seeing the same thing with the neurons.
Thank you so much. We have one more very quick question. I think an audience asked, does those VMPO neurons express TOL4, which is a receptor that can directly sense LPS? So whether or not the neurons respond to LPS directly and without this pro-inflammatory cytokine pathways.
Yeah, so I didn't do this. So I did look in the single cell data. I didn't see any TLR4. But there is TLR4 in the brain, right? Because the brain is able to somehow sense that there's bacterial infection. Bacteria can infect your brain. So that is the case. But I didn't see it in the single cell data, and I didn't look using the in situ experiments. So I don't know if these cells do. I wouldn't be surprised if I did. I think the TLR4 expression is pretty wide, actually, because as soon as there's a bacteria in the brain needs to know. So. But I should say that the LPS doesn't, there's mixed data on whether LPS crosses the blood-brain barrier. I've now heard more recent data showing that it does. So anyway, yeah.
Thank you so much. That's beautiful. We still have many questions in the chat. If you have time, please go through, and then we will try to see if we can find a time to discuss. Thank you so much. Thanks. And Jainu, please take away for our wonderful second speaker.